So in this video we're going to do a detailed review of mitosis, and briefly, mitosis is just the process of cell division. And we start with interphase, and as you see, here's our cell, and those are the spindle fibers and centrioles, and that's our nucleus. And then we move to the second phase, which is called prophase. And in the prophase, as you see here, the centrioles are moving to the opposite sides, because in the end there's going to be a centriole and spindle fibers on this side, and a centriole and spindle fibers on this side, and it's going to pinch in the middle, and we're going to have two separate cells. So what prophase is it's when the chromatin condenses into chromosomes. Right here, as you see here, there is no chromosomes yet. I'm going to zoom in on that. See, the chromosomes usually are in X shapes, like right here. Here the chromatin hasn't condensed yet. So it condenses, then the cell divides, and we have two new cells. So here the chromatin condenses and the centrioles separate. Then the spindle fibers begin to form. So the nuclear envelope begins to break down. Now we move to the metaphase, M. The metaphase, here the chromosomes line up across the center of the cell. And each chromosome is connected to the spindle fibers at its centromere. So as you see right here, let me zoom in. See, the chromosomes, they broke out of this envelope, the nuclear envelope, and now they're all strewn in the middle. And right here, the reason why they're lined up in the middle perfectly, because soon it's going to pinch, and then they're going to separate, and we want an equal amount, sort of almost like an equal amount of chromosomes in each cell. And we move to the anaphase. The anaphase. Here we have it's very similar to the metaphase, but here the chromosomes, they split apart. And now we're going to start pinching this way. So when cytokinesis kicks in, the cell would start to pinch by itself like that, and it'll get smaller towards the middle, and then it's going to separate. So in the anaphase, the sister chromatids separate into individual chromosomes, and they're moved apart. Now we move to the telophase, telophase, and here the chromosomes gather at the opposite ends of the cell and lose their distinct shapes. And now two new nuclear envelopes will form. As you see here, that's a nuclear envelope, that's a nuclear envelope. See how inside the nuclear envelope we have our chromosomes. They haven't decondensed into chromatin. They're still chromosomes inside. As you see, it's beginning to pinch. And moving on, cytokinesis. Cytokinesis is our last phase, our last step to this process of mitosis. As you see here, the cytoplasm pinches itself. See, that's the cytoplasm, that outline right here. Not really the outline, it's just this inner outline, all that stuff inside. So the cytoplasm begins to pinch itself in half. And then each daughter cell, what they mean by daughter cell is like the products of this. It's like here we have our parent, and then we end up with like the daughters. And here each daughter cell has an identical set of duplicate chromosomes. So, yep, each cell has the same type of chromosome, DNA, because in the end, your same body, all the DNA that is carried within your cells is identical. So, in summary, here we have PMAT, and a good way to remember the M, metaphase, is that M for middle, they're all lined up in the middle. That's a nice way to remember metaphase if you have a quiz. And that wraps it up for mitosis.